Hi, everyone. Welcome back to the podcast. Today, we'll be covering Chapter 6 of Lori Lundy Ekman's Neuroscience for Rehabilitation, 6th edition. The chapter is called Neural Communication, Synaptic and Extrasynaptic Transmission. If you've already heard this lecture in person, think of this episode as your brain's reuptake inhibitor, slowing down, forgetting, and keeping neurotransmitters in the synapse just a little longer. So grab your calcium ions and let's jump in. What is a synapse, really? Let's start with the synapse, which is basically the neuron's version of personal space. A synapse has three parts. First, we have a presynaptic terminal. This is the sender of information. Then we have a postsynaptic terminal, which would be our receiver. And then there's a synaptic cleft, kind of the awkward gap between them. And that postsynaptic cell, it could be another neuron, muscle fiber, gland, or even a blood vessel. Neurons are very versatile communicators. You'll also hear about types of synapses. We have an axodendritic, which is axon to dendrite, axosomatic, which is axon to soma or cell body, and axoaxonic, which is axon to axon. Yes, neurons can talk to each other in a lot of different ways. Apparently, they're very social, but they like to keep it complicated. Now let's talk about neurotransmitter release, or as I like to call it, the calcium party. Here's how communication actually happens. An action potential arrives at the presynaptic terminal. That depolarization will open voltage-gated calcium channels, and calcium ions rush in. Calcium then binds to docking proteins, which cause a synaptic vesicle to fuse with the presynaptic membrane and release neurotransmitters into the synaptic cleft. Neurotransmitters then diffuse across the synaptic cleft, binding to receptors on postsynaptic membrane, and those receptors change shape. That shape change could either open an ion channel or activate intracellular messengers. In translation, the message has been delivered. How do we end the conversation? Here's how they regulate the transmission. Neurons are efficient communicators, but they won't be chatty forever. Synaptic transmission can be regulated by reuptake transporters, pulling neurotransmitters back into the presynaptic terminal. Enzymes are present to break down any neurotransmitters that are left in the cleft. They may also diffuse away from the synapse, and there are receptor shutoff mechanisms, including receptor endocytosis, where part of the postsynaptic membrane folds into the cell, leaving fewer receptors on the neuronal surface that can be activated. This is all just termination of the initial signaling. So EPSPs and IPSPs, to fire or not to fire. When neurotransmitters bind to receptors, they create postsynaptic potentials. These are potentials acting on the receiving end of the neuron. EPSPs are excitatory postsynaptic potentials, and they cause a local depolarization, so an increase in polarization. IPSPs are inhibitory postsynaptic potentials. These will cause a temporary local hyperpolarization, so going down in polarization. Remember, these changes are local. They are not full action potentials, but they can have an influence on whether or not a neuron will fire under the right circumstances. Now let's discuss presynaptic facilitation and inhibition. So what's going on the sending side of the neuron, or presynaptic? Sometimes neurons can help or sabotage each other. Presynaptic facilitation happens when one axon slightly depolarizes another axon terminal, causing more neurotransmitter release. Presynaptic inhibition happens when one axon slightly hyperpolarizes another terminal, causing less neurotransmitter release. Same synapse, very different outcomes. Neurotransmission comes in two speeds, fast versus slow. So fast transmission takes milliseconds, very, very quick. They act directly on the ligand-gated ion channels and cause rapid changes in membrane potential. Slow transmission can take hundreds of milliseconds to minutes or days for transmission. They often involve extrasynaptic or volume transmission, axon G protein coupled receptors or GPCRs, and the effects last much longer. Fast is like texting. Slow is like that old pen pal letter you sent to your second grade bestie in 2002. Did she even ever get it? Now we can discuss G proteins and second messengers. So when neurotransmitters bind to G protein coupled receptors, GPCRs, G proteins are activated. These G proteins can open ion channels, activate second messengers, trigger gene activation, release internal calcium stores. Second messengers amplify signals and regulate systems involved in mood, pain, perception, movement, motivation, and cognition. When this system malfunctions, it's implicated in conditions like epilepsy, mental illness, Alzheimer's disease, Parkinson's disease, Huntington's disease, multiple sclerosis, and chronic pain. So yes, it is very important for this process to work properly. So receptors are more than just a neurotransmitter. A key takeaway is that the effect of a neurotransmitter depends on the receptor it binds to and how it's released, not just the neurotransmitter itself. For example, acetylcholine, or ACH, causes fast transmission in the neuromuscular junction, but also induces slow transmission in the heart. Same chemical, different receptor, totally different effect. Now let's talk co-transmission. Neurons can release more than one neurotransmitter at the same synapse, called a co-transmission. Often, one transmitter is released during low-frequency firing, and another is released during high-frequency firing. Basically, the neurons escalate when needed. Let's get into the specific neurotransmitters. First, acetylcholine, or ACH. This is a major transmitter in the peripheral nervous system. It's used by all lower motor neurons to skeletal muscle and acts on nicotinic and muscarinic receptors. Clinically, myasthenia gravis destroys nicotinic receptors at the neuromuscular junction. Also, a loss of ACH neurons is the hallmark of Alzheimer's disease. Now, glutamate. Glutamate is an amino acid neurotransmitter. It is the primary excitatory neurotransmitter in the central nervous system. It's essential for learning and development. It acts on ionotropic receptors and metabolic glutamate receptors, or mGlurs. Abnormal glutamate activity is linked to excitotoxicity, epilepsy, chronic pain, Parkinson's disease, schizophrenia, and stroke. And let's talk about GABA. So GABA is a major inhibitory neurotransmitter. GABA receptors are chloride channels. GABA alpha receptors are chloride channels, and GABA beta receptors are G protein coupled receptors, GPCRs, and they are linked to potassium channels. Benzodiazepines and barbiturates are medicines that are able to enhance GABA's effects and are used for things like anxiety, sedation, and seizures. Glycine receptors have inhibitory chloride channels in the spinal cord, brainstem, and basal ganglia. They are also required for NMDA receptor activation, making it important for learning and memory. Now we have our amine neurotransmitters. So dopamine is motor activity, cognition, and behavior. Norepinephrine has an effect on attention and surveillance. Serotonin has an impact on mood, sleep, arousal, and cognition. So when you're looking at these three collectively, dopamine, norepinephrine, and serotonin can help regulate our emotion, cognition, and overall mood. Another amine neurotransmitter is histamine. and it's concentrated in the hypothalamus. When considering peptides, nitric oxide, and endocannabinoids, here's information that you need to know. Opioid peptides reduce pain perception. Substance P and calcium gene-related peptides are involved in pain signaling and migraine. Nitric oxide diffuses directly into the cells and affects long-term neural changes. Endocannabinoids travel backwards across synapses to limit neurotransmitter release. Yes, these neurons can text backwards. There are types of drugs that impact our neurotransmitters, and they can act as agonists and antagonists. Agonists will mimic the job of the neurotransmitter. Antagonists will block neurotransmitters' effects or release. Many drugs work by targeting these mechanisms. Now, there are quite a few disorders that impact synaptic function. Myasthenia gravis is an autoimmune condition in which antibodies destroy nicotinic receptors on muscle cells. Lambert-Eaton syndrome typically occurs in people with small cell lung cancer, where antibodies block presynaptic calcium channels, which eventually causes decreased excitation of the muscles. And channelopathies is an ion channel dysfunction that may cause epilepsy, migraines, or paralysis. The final takeaway for this is that synaptic communication really depends on what neurotransmitter is released, how the receptor is activated, and whether the transmission is fast or slow. Neurons don't just talk, they regulate, modulate, amplify, and adapt. That wraps up chapter 6. If your neurons feel slightly more connected than before, the synapse has done its job. Thanks for listening, and remember, communication is everything, even at the cellular level.